Hello everyone. So the next topic which we will be discussing is about the biosynthesis. Now what do you understand by the term biosynthesis? So biosynthesis, it is an enzymatic process where simple compounds are used to synthesize the macromolecules inside the living cell. So this is what do you mean by the process of the biosynthesis. So let's start with it and discuss it in a little bit more detail. So introduction. So biosynthesis, it is an enzymatic process where the simple compounds are used to synthesize the macromolecules inside the living cells. So it is a multi-step process. So what is it? It is a multi-step process. The products of biosynthesis are essential. What are they essential for? They're essential for the cellular activity, growth, development, and survival of the living organism. So these are the essential things which are being performed by the process of the biosynthesis. It is an anabolic process of metabolism. So examples include the sugar biosynthesis, which is by the Kelvin cycle, or gluconeogenesis, then protein synthesis by the translation, fatty acid, lipid, and the cholesterol biosynthesis. So we will be discussing each of these biosynthesis in detail now. So biosynthesis, it is a major scientific research area where the synthesis is done in vitro using the chemicals and also by the recombinant methods in the organism using enzymes and the substrates. Now, what are the key features of the biosynthesis? So biosynthesis refers to a anabolism. So it was being stated in the previous slide as well that it is an anabolic process. So it refers to the anabolism. Then comes, it is a multi-step and multi-enzymatic process. It occurs in one or single multi-cell organelles. So biosynthesis is an energy-driven process. Chemical energy is utilized in the process, which we get when the bond is being broken. So example, there is a terminal high energy phosphate group which is being hydrolyzed from the ATP energy. Then there are multiple enzymes which are involved in the process. Enzymes catalyze the reaction by decreasing activation energy, thereby increasing the rate of the reaction. There are many enzymes which require the cofactors for proper functioning, such as the prosthetic group, example metals and non-metals, ions and coenzymes, and the examples here will include the NAD, NADP, FAD. So these are some of the examples. Now comes the macromolecules, which are generally the polymers of monomers. So the synthesis of the monomer is an important step. Example, amino acids for protein synthesis, nucleotides for the DNA and RNA synthesis. So biosynthesis, it is controlled at each step and there are many enzymatic activities that are regulated by a feedback mechanism. So this is what is the features of the biosynthesis. The next comes is the sugar biosynthesis. So it is an important step for the living organisms. So most of the energy comes from the food from the plants. So most of the energy comes from the plants only. So being the primary producer, what do plants do? So plants, they fix the atmospheric carbon dioxide to form the organic compound such as the sugar. So there are two main pathways that are being involved, that is the Kelvin cycle and gluconeogenesis. So the Kelvin cycle is the carbon fixation, reduction of C from CO2 to triose phosphate, a sugar. Then the gluconeogenesis, which is the formation. What is it? It is the formation. Gluconeogenesis, as the name suggests, so it is the formation of glucose from non-carbohydrate precursors such as the lactate, the glycerol, and the pyruvate. So the Kelvin cycle we will be discussing in somewhat detail. So Kelvin cycle. The plants and there are some other microorganisms which fix the atmospheric carbon to synthesize the sugar. So what is Kelvin cycle? Kelvin cycle, it is a light independent or the dark reaction of the photosynthesis. It occurs in all the photosynthetic organisms, that is the C3, C4, CAM. So it is not directly dependent on the light, but it utilizes the ATP and NADPH produced in the light reaction. Now it is a three-step process: first, second, and third. So the first step includes the carbon fixation, which is also known as the carboxylation. Second comes the reduction, and the third is the regeneration. So the first is the carbon fixation. It is the first step here where CO2 is fixed by a keto-pentose sugar phosphate, that is RUBP, to form the 3-PG, that is the 3-phosphoglycerate, and the enzyme Rubisco, it is being used to catalyze the reaction. Next comes is the reduction. So reduction, it involves a series of steps which reduce the 3-PGA to triose phosphate such as G3P and DHAP, which are further converted into fructose, glucose, starch, or sucrose. The last step is the regeneration. So in this last step, RUBP is regenerated back to continue the cycle. So this is what the Kelvin cycle is about. The next comes is the gluconeogenesis. So what is it? It is a process of sugar synthesis from non-carbohydrate precursors. So it is present in microorganisms like fungi, plants, as well as some of the animals. So in this particular process, what is happening is substrate coming from the breakdown. It's coming from the breakdown of proteins, lipids like glycerol, lactate, amino acids, which gets converted to pyruvate and then it gets converted to glucose. So this is what is known as the process of gluconeogenesis. The next comes is the protein biosynthesis. So this particular process takes place by the process of translation from mRNA. The genetic information which has been contained in the DNA, it gets translated into the polypeptide chains, which are then folded to form the proteins. So the proteins, they are the polymers of the amino acids, and the amino acid sequence in a polypeptide chain, it is being governed by genetic codes which is present in mRNA. So it is a three-step process. Now what are the three steps here? The three steps here is the initiation, elongation, and the termination. So the protein biosynthesis is a three-step process. The next is about the biosynthesis of the amino acids. So there are about 20 amino acids which are known, which are a part of the protein structure, and humans, they do not have the capability to synthesize all the amino acids in the body. So amino acids can be synthesized, that can be synthesized in a body is called as the non-essential amino acids, and that those have to be supplemented through the diet, they are called as the essential amino acids. Now the non-essential amino acids are synthesized from various intermediates of metabolic pathways such as citric acid cycle. Some of the examples of this includes the many amino acids which are synthesized by the alpha keto, that is synthesized by the alpha keto acids and then transmitted to form other amino acids: glutamate, glutamine, arginine, proline, and that starts with the alpha glutarate, which is an intermediate in the citric acid cycle. So this is what is the biosynthesis of the amino acids. The next comes is the biosynthesis of the nucleic acid. So DNA, it is a polymer of nucleotides which is being joined by the phosphodiester linkage. DNA, which is a double helix structure, it has two strands of DNA that is being joined by the hydrogen bonding between complementary nitrogenous bases. So DNA synthesis takes place in the nucleus. DNA replication is a semi-conservative process, and DNA polymer catalyzes the reaction. Whereas if I talk about the RNA, it is being synthesized from the DNA by the process which is known as the process of transcription. So the genetic information gets in DNA, gets transcribed by the RNA, and RNA synthesis is being catalyzed by the RNA polymerase. So DNA, RNA, they are the polymers of the nucleotides which are made up of nitrogenous bases, pentose sugar and phosphate. The next comes is the biosynthesis of the fatty acids, or you can see the biosynthesis of the lipids. So the biosynthesis of lipids has been divided into biosynthesis of fatty acids, biosynthesis of triglycerides, and biosynthesis of the phospholipids. Now, lipid, which is an important constituent of the cell, it is made up of a lipid bilayer. So energy is stored in the form of lipids, and they are also important in signaling. So there are many hormones that are made up of lipids. Fatty acids, which are the simplest type of lipids, triglycerides, phospholipids, glycolipids, cholesterol, they all are the lipids. So if I talk about the biosynthesis of the fatty acids, then it takes place in the cytoplasm. Fatty acids, they are being synthesized from the acetyl coenzyme A using NADPH, which is an intermediate of the glycolytic pathway. So the enzyme fatty acid synthase catalyzes the reaction. The next is about the biosynthesis of triglycerides. So the three fatty acids which combine with the glycerol to form the triglycerides. So it is commonly known as the fat. The next is about the biosynthesis of phospholipids, which is an important constituent of the cell membrane. They constitute the lipid bilayer surrounding cell organelle. So phospholipids, they are amphipathetic and they have a hydraulic or polar head, which they have is. The next is about the cholesterol biosynthesis, which is a type of lipid known as the sterol. So it is synthesized in the body from the acetyl coenzyme A. And most of the cholesterol synthesis takes place in the liver and the intestine. So this is about the biosynthesis of the fatty acids. So this was all about it. These are some of the references.